Hello students, today I am here to discuss regarding the complement system. Now we will discuss in details today about the following topic. What is complement system? What are the components and function of complement system? What are the different pathways of complement activation? Difference between classical and alternate pathway of complement activation and disorders of defective complement system. So let us them in details. Now the complement system. Complements are the part of immune system that enhances or complements antibodies and phagocytic cells to clear pathogens. What does it mean? Actually, complement is the name given to a system that consists of some non-specific proteins, particularly they are glycoprotein chemically, and they are present in the normal human serum. They have got the ability to lyse or damage cells, the foreign cells which may be a bacterial pathogen, a virus cell, tumor cells, and to stimulate antigen-antibody reaction. Actually, it is activated by the antigen-antibody complex and therefore acts consequent to and it supports the immunoglobulins in the adaptive immune system. That is, it complements the action of antibodies in adaptive immune system. Thus, it is known as the complement system. Now, as I have already defined that they complement or support the action of immunoglobulins. The complements were initially thought to be a part of adaptive immune response, but it is seen that complement system in some way also take part in the innate immune response. Now, let us have a look in the historical milestones in the identification of this complement system. In the year 1888, George Nuttall found that the sheep blood have some protein that can kill the pathogen, the bacteria Bacillus anthracis, causing anthrax in the sheep population. But when the serum was heated, this property was lost. In the year 1891, Hans Ernst August Buchner gave it the name alexin, and he also find the same property that some components are there in the serum which has heat labile property and they has some action to kill the pathogens. Then Jules Baudet of Pasteur Institute even found the same property in the serum, and he found two type of components in the serum. One is heat labile, another one is heat resistant. In the year 1899, Paul Ehrlich named this heat labile component as complement. Now, what are the properties of this complement proteins? This complement proteins has an astonishing similarity with the coagulation proteins. That is, they need to be activated. If you recall the coagulation cascade in the intrinsic or extrinsic pathway, they need to be proactivated by the phospholipids and they are in the protease or zymosin form and then they are activated. Similarly, the complement proteins are also present as inactive zymosin. And what is another similarity with the coagulation cascade? Coagulation cascade has an astonishing amplification activity. Similar type of property is also seen in the complement cascade. That's why this complement cascade were thought to be a reminiscent of the coagulation cascade proteins. Now, these complement proteins, as I have said, they are biochemically glycoproteins. They are synthesized by the liver cells, hepatocytes, macrophages, monocytes and the epithelial cells of the gastrointestinal tract. They are present as inactive form, that is proprotease or zymosin in the circulation. Consists of 30 proteins and contribute to 3 gram per liter of the total proteins. What are the important functions of the complement proteins? They help in cell lysis, that is, they degrades the foreign cell. Opsonization means making the foreign cell tastier or coated with the antibody. Chemotaxis, as we will discuss regarding the various type of complement protein, we will see some of the complement proteins are chemoattractants and they help in agglutination. So the important functions of the complement system are cell lysis, opsonization, chemotaxis and agglutination. Now let us see what are the various pathways of the complement activity. The complement system were initially thought to have two pathways, that is the classical pathway, the alternate pathway. But recent advances have delineated another pathway, that is the lectin pathway. Now let us discuss each of the pathway in details. Firstly, with the classical pathway, begins with antibody IgM or IgG binding to a cell surface and ends with the lysis of the cell. Proteins in this pathway are the C1 to C9. Why these complement proteins are named as C1, C2 and C3, C9? In the order they were first discovered or identified, not in the order of the reaction. When a complement is activated, as I have said, it is a proprotease, and when it is activated, it gets splitted into two parts. The smaller fraction, which is denoted by the small a, and larger part, which is denoted by the capital B. For all of them, the larger part is the capital B, except with the factor 2, C2, though B is the active form, but it is smaller than the A fragment in cases of C2. Now the complement cascade consists of three sub-pathways, that is, initially we need to activate. So the activation phase, the amplification phase, and lastly the attack phase, where a membrane attack complex is formed and it causes lysis of the pathogen or the foreign cell. Now this is the diagram or the schematic representation of the classical pathway. In the classical pathway, the complex system is triggered when an antigen-antibody complex binds to and stimulates the protease activity of the factor C1. So you can see this is the foreign cell and the Y-shaped antibody is bound. Now what happens when the C1 is cleaved? It is cleaved into three fragments, S, Q and R, and the C1 then cleaves the complement factor to form the two smaller proteins C2A and C2B. The C2A and C2B cleaves the complement factor C4 to form C4A and C4B. Now the C2A and C4B, as I have said in my previous slide, 2A for all other practical purposes, the A fragment is the smaller, but for the complement to the A fragment is larger in size. So this the heterodimer C4B and C2A activates a protease that is known as C3 convertase. This C3 convertase activates or cleaves the C3 into two fragments, C3A and C3B. The C3A has an anaphyllotoxin-like property. As you can see, The C3A binds to the receptors on the basophils or the mast cells, and we know the granules of the mast cell triggers to release the vasoactive amines like the histamine, which causes vasodilatation and increases the permeability of the vasculature and thus triggers the anaphylaxis response. Now this C3A now binds to the 2A2B heterodimer to form the heterotrimer, and it cleaves the C5 into C5A and C5B. The C5A is also a potent anaphylatoxin and it is a chemoattractant for the neutrophil. Now, as you can see here, we have a complex which has the C4B, 2A3 and C5B. Now this complex will activate the other complexes, that is 6, 7, 8 and 9 to form the membrane attack complex. This membrane attack complex kill the bacterial invaders by binding to and opening a pore in the plasma membrane. Following lysis, you can see here the pores in the plasma membrane are formed. Following the lysis, the bacterial remains are cleared off by the macrophages with the help of their phagocytic properties. So this is the classical pathway. The C3A and C5A protein serve as chemoattractants and they are anaphylotoxins. They also have another role. They recruit the leukocyte to the site of infection and stimulates the inflammatory response. Now, as I have said initially that the classical pathway has three subphases: activation, amplification and finally the formation of membrane attack complex. So, let us see the second phase, that is the amplification. The C1 complex is again cleaved into three parts: Q, S and R. Each C1S creates many C4B and 2A fragments. Now this heterodimer creates, if it is X, it will create 2X form of C3B, and C3B again creates 4X concentration of 5A and finally it will be 8X concentration of membrane attack complex. As you can see, there is a tremendous geometric or exponential rise of the complement factors, just like the coagulation cascade, and this is the amplification phase of the complement activation pathway. Now the factors which influence the complement lysis are the optimum temperature and the pH, immunoglobulin class and subclass. As we have seen in the classical pathway, it is triggered when the antigen antibody complex binds to and then it stimulates the protease activity of the C1 complex, antigen density and flexibility at the hinge region of the antibody complex, because we know that the antibody binds to the antigen by some specificity on specific receptors that are paratopes upon encountering a novel antigen. So these are the factors that influence or rather that alters the complement lysis: the temperature and the pH, immunoglobulin classes like IgM and IgG, the antigen density and the flexibility at the hinge region of the antibody. Now the second pathway is the alternative pathway. This alternative pathway is also known as ticking over. Why? Because the complement system involves the activation of the factor C3 by chemical hydrolysis. In the classical pathway we have seen we require an antibody to activate the complement pathway, but here it is activated by the chemical hydrolysis. The C converts C3 to C3b. Normally, C3b is very short-lived and quickly inactivated by proteins on the surface of body's own cells. However, bacteria or foreign material may lack this surface protein, allowing the complement C3b to bind and stay active. Then factor B binds to the C3b and B is then cleaved by factor D into capital B, smaller fragment A and another fragment B. The C3b and the B fragment of the factor B acts as a C3 convertase, and this C3b and B fragment of the factor B again acts as a protease for C5 activation, and then this E5 is cleaved to 5A and 5B, and then it follows the classical pathway involving the 6, 7, 8, 9 to form the membrane attack complex. So, in the alternative pathway, we need two another proteins, factor B and factor D. The factor D is required to cleave the factor B into smaller fragments A and B. The B fragment remains attached to the C3 and acts as a C3 convertase, and this C3b and B fragment of factor B, it acts as a protein convertase of the factor 5. So this is the activation of the alternative pathway. This is the classical diagram. Here you can see factor C3. It is cleaved to C3A and C3b. As I have said earlier, C3 is an anaphylotoxin. This is chemically activated. Now this C3b will bind to the factor B, and this whole complex will bind to factor D. Once factor D is bound, this will cleave the factor B into smaller A and smaller B fragment. The A fragment will go on, and this whole again will amplify the C3 convertase activity. The C3 will again be cleaved to 3 and 3B. Now this will again activate the C5 convertase. The C5 will form the membrane attack complex, and this will cause lysis of the cell. This is the newer pathway which is known as the lectin pathway. This has certain similarity with the classical pathway, but there is a major difference that this does not require any antigen antibody. As we have seen that binding of antigen to the antibody is a prerequisite for the activation of the C1 to QRS complex. Here we do not require any antibody. What are lectins? Lectin refers to proteins that bind to any polysaccharide. Now this lectin pathway wherein the complex are formed when a complement factor which is known as mannose binding lectin or mannan binding protein binds bacterial polysaccharide to generate a complex that activates C4. This mannose binding lectin or mannan binding protein belongs to the collectin family of proteins that are synthesized by the liver. The multimers of this mannose binding lectin forms a complex with the MSP1, 2 and 3. These are quite similar to the C1, R and S molecules of the classical pathway. And this mannose binding lectin binds specifically arranged mannose residue on the surface of the pathogens. They are or the lipopolysaccharides which are present on the gram-positive bacteria, even in some fungus, and MASP1 and 2 are activated to cleave the components of C4 and C2 into 4A, 4B, 2A and 2B. This is a diagram of the mannose binding lectin. This mannose binding lectin has this, as you can see, This are the heterotrimers, green colored, which binds the carbohydrate domains, that is the mannose residue. And the core of the trimer is formed when three collagen-like domains intertwine to generate an extended tail that leads to the globular heads which contain the carbohydrate binding domains. This and the actually this is a, as I have said, lectins are what? They are proteins. So this protein will have one amino terminal and it will have one carboxy terminal. The amino terminal has a collagen like domain and the carboxy terminal has the carbohydrate binding domain. So this carbohydrate binding domain will bind to the mannose or the lipopolysaccharide residues which are present in the cell wall of the gram-positive bacteria, viruses or fungus, and this collagen-like residue, they will bind to the remain attached to the native cell. So this is the mannose binding lectin. Now this is a common question which you will face in Viva, that what are the differences between the classical and alternate pathway. The classical pathway represents the adaptive immunity, whereas the alternative pathway represents the innate immunity. It requires an antibody, more specifically IgM, that is antigen antibody complex is required to activate this factor. C1 does not require any antibody. The C3 is activated by chemical hydrolysis. Requires all the factors. It requires factor C3, then factor C5 to C9. Does not require factor B, factor D, whereas the alternate pathway requires the factor B and factor D. So this is the difference between the classical pathway and the alternate complement pathway. Now, these are the complement associated diseases. The C1 esterase deficiency can cause hereditary angioedema. That means the patient will have angioedema if this due to attack of anaphylotoxin. Next is low complement level are found in systemic lupus erythematosus. This is an autoimmune disease and this patients will have low complement level. Paroxysmal nocturnal hemoglobinuria. This is an autoimmune hemolytic anemia. Here you will find that the patients have defect in the cell surface decay accelerating factor. And low complements are associated with autoimmune glomerulonephritis. That's why the estimation of complement factors are given in cases of autoimmune nephritis. The C3 and C4 factors, they are estimated quantitatively in the laboratory using nephelometry. These are the common diseases and these questions are many times repeated in postgraduate entrance examination. Which enzyme deficiency causes hereditary angioedema? Paroxysmal nocturnal hemoglobinuria is associated with which protein deficiency? So these are the common questions. So to sum up, what are complements? They are heat labile glycoproteins that are present in the human sera that complements the activity of the antibody to cause cell lysis. Initially, they were thought to be a part of the adaptive immunity, but with the advent of the classical alternative pathway, they have some role in innate pathway also. This complement are almost 30 proteins, but in the cascades, classical we have or the alternative, we have found that the C1 to C9 are more important. Why they are so named? In the order of the discovery. They are synthesized by the liver cells, monocyte, macrophages, intestinal epithelial cell. What are their important functions? Cell lysis, opsonization, agglutination, and in the killing of the pathogens. What are the three important pathways? classical pathway, alternative pathway, and the Mannose-Lectin binding pathway or the lectin pathway. The differences between the classical and alternative pathway: classical is a part of the adaptive immunity, alternative is a part of the innate immunity. Classical pathway required antibody, specifically IgM. Alternative pathway, chemical hydrolysis of the C3, involves all the factors in cases of classical pathway, where it is C3 and C5 to 9 in alternative pathway. We require other proteins in cases of alternative pathway C, that is factor B, factor D, whereas these factors are not required. Various diseases like SLE, autoimmune glomerulonephritis, hereditary angioedema, they are caused due to defective complement cascade, and we need to uh, go for quantitative estimation if we suspect such a disease. Thank you, and thank you to this rabbit here. All of the immunological experiments, most of them have been done from the poor soul of these rabbits, and they have helped us to know our immune system clearly. If you have any question, you can mail me. Thank you.